Welcome everybody to today's webinar. So in the next 30 minutes approximately, I would love to introduce you to new solutions for non-viral T-cell engineering, which offer a great potential to advance T-cell based cell and gene therapy. So I'm an employee of Miltenyi Biotec, and those are the disclaimers mainly around the products that we are providing. So during today's webinar, I would like to introduce you to the cell and gene therapy market. I think you're very much familiar with that. Then I would like to focus on different gene editing technologies and the connected challenges. Then after that, I would like to propose some solutions for those challenges. And last but not least, I would like to show you some very impressive publications from our collaboration partners who used our Clinimax Electroporator. The cell and gene therapy area, as you are probably very much aware of, has really shown remarkable growth rates in the last years. So when we look at the number of preclinical projects or clinical trials, these numbers have really increased dramatically in the last years. And when we look at cell therapies, we can actually divide it into non-genetically modified cell therapy projects and the gene therapy pipeline. And what we can see is that there is a clear trend towards the gene therapy pipeline. So they are showing an even more impressive growth than the non-genetically modified cell therapy projects. And of course, within that gene therapy pipeline, we will also find all the gene-engineered cell therapies. Focusing on approved gene and cell therapy drugs, we can see that there are already 16 approved drugs in the gene therapy area, 8 of them being actually engineered cells, 3 of them being engineered hematopoietic stem cells, and 5 of them being engineered T cells. And I think we all are aware of these 5 commercially approved CAR T cell products already. And what we also are aware of, I think, is that there are already quite a lot of companies with disclosed CAR T or TCR assets in their preclinical pipeline. And when we are just looking at CAR T cell clinical trials, there are already more than 1,000 of those ongoing. And looking at the annual growth rate, which is more than 20%, we can really see that the engineered T cell area is really a main driver. But even with 5 commercial CAR T cell products already being on the market and having been developed successfully, I think we shouldn't forget how long the path was to commercialization. How many obstacles and hurdles were on that way and still are if thinking about developing a new cell and gene therapy. So what are those challenges and obstacles? I think we can easily divide them into 3 sections. One is the manufacturing, one IPC/QC, and then of course the treatment performance. So let's start with manufacturing. I think one thing that really sticks out is the cost of goods. And within the cost of goods, I think we're all very much aware that viral vectors are really a key cost driver. Then scaling up, automation, standardization is also usually an issue and a challenge to overcome. And last but not least, translation of your protocol into a GMP compliant fashion is always a challenge. Then very closely connected to the manufacturing is the IPC and QC, and their major challenges are the assay development, having a standardized and robust assay to be able to deal with different starting products and also different end products, because depending on the patient, those products might really vary a lot. And then last but not least, the treatment performance. So once we manage to successfully manufacture our cell and gene therapy drug product, that still doesn't mean that it will be efficient in the patient. So efficiency is still a hot topic, especially when we are talking about CAR T cells and solid tumors. I think there is still a lot of room for improvement. Also when looking at specificity or also safety. So normal side effects of CAR T cells are CRS, for example, or even neurotoxicity. So also there is still a lot of room for improvement. So what could be general approaches to tackle those challenges? When we are thinking about manufacturing, and we already mentioned the high cost of goods, especially the costs for the viral vector, one idea would be to switch to a non-viral gene engineering strategy to avoid the high costs. Then it's always good to start already early in your development with an automated and closed system that makes translation into GMP environment much easier. And it's also very advantageous to start with a scalable system, also to make the translation easier. Then, when thinking about IPC and QC, it's very good to benefit from existing workflows, to start off with high quality products, and also to rely on robust and proven products. When thinking about treatment performance, the good news is there are actually already a lot of impressive projects out there to circumvent side effects and increase efficiency and so on. However, most of them actually require complex gene engineering approaches. And on the next slide, I would like to show you some of these promising projects. So as just mentioned, engineered T-cell products in cancer therapy still face a number of challenges, for example, related to safety or efficiency, or also decreasing the vein-to-vein time, or also decreasing costs. What you can also see in the table is, like mentioned, there are already a lot of approaches out there, a lot of promising projects to tackle those challenges. And this list is definitely not complete, it just lists a few of these very promising approaches. What also becomes quite clear when you look at this list is that the complexity of gene engineering is quite high. So for example, if we look at the allogeneic approach, where the idea is to create a universal CAR, the endogenous TCR knockout is one of the major strategies, and that requires quite complex gene engineering. And this complex gene engineering, of course, once it proves to be successful, then of course needs to be translated into clinics, and that is usually a major challenge. And then apart from cancer, of course, there are also other indications where T-cell engineering might also be very promising. So for example, in HIV. So there are also already approaches there, for example, targeting CCR5 or targeting CXCR4. Having just seen all these impressive new approaches, I think we all realize that non-viral gene engineering might not just be a key factor to lowering your manufacturing costs, but they might be also the door opener to advance cell and gene therapies completely and bring them to a new level. From a technical perspective, there are several different gene editing technologies out there with two major strategies. One would be the viral strategy, using either integrating viruses like lentivirus or retrovirus, or using non-integrating viruses like, for example, adeno-associated viruses. The second major strategy is the non-viral strategy, also either with integrating systems like transposons. Examples would be Sleeping Beauty or Piggyback, or non-integrating systems like plasmids, RNA, designer nucleases, for example, TALENs or CRISPR-Cas. But please keep in mind, when working with non-viral strategies, there's always an additional gene delivery technologies required, because somehow the genetic information needs to be transferred into the cell. And one very widely and well-established method for that would be electroporation. Looking at real-life usage of gene engineering approaches, it's really surprising to see that 88% of the approaches actually use viral vectors for their gene engineering. And the preferred viruses are lentivirus and adeno-associated virus. And this is really surprising, because actually there is a huge potential for non-viral gene engineering to advance cell and gene therapies. It enables specific gene and base editing, and therefore it enables specific knock-ins and also knockouts. It's offering an unlimited promoted choice and reducing not only your cost of goods, but potentially also your safety precautions. And also all the different technologies that I just showed on my last slide, so using TALENs, CRISPR-Cas, and so on, this is all enabled with the non-viral gene engineering. So there is still a very huge unused potential to advance cell and gene therapies, while using non-viral gene engineering strategies. So how can we help you to use that unused potential? And first of all, of course, who are we? So Miltenyi Biotec was founded in '89, and nowadays we have more than 3,500 employees worldwide. About a quarter of these employees are working in our research and development. And more than 46,000 publications are actually using our products, and we have more than 17,000 products on the market already. More than 100,000 cell therapy procedures were done with our products, and per year, about 6,000 patients are treated with cell products that have been manufactured using our technologies. So you can clearly see that even having been founded as a research company, we are more and more going into the clinical direction. To underline that, I would like to show you how we became the clinical T-cell engineering experts. So it all started in 2012 when we launched the CliniMax Prodigy. This system is used for automated and closed manufacturing of cell therapies. Then, 2 years later, we initiated the CAR program, and after another 2 years, in 2016, we released the CliniMax Prodigy TCT process, which is used for CAR manufacturing. Then, in 2017, the cell factory in Bergisch Gladbach and another manufacturing site in Sunnyvale, California opened. And in 2018, we were granted a generic manufacturing authorization for CAR T cells. The same year, we produced our first trial patient product, and we extended our IPC and QC portfolio by releasing the C19 CAR T-cell Express Mode package. 1 year later, in 2019, we also released our first C19 CAR detection reagent. For now, we have a couple more, and we successfully transferred our manufacturing protocol to an external CMO, and they produced their first cell product. And now, in 2021, I'm very happy to announce the release of the CliniMax Electroporator and the corresponding software, which is called T-cell engineering. And that system will not only allow the viral transduction of T cells, but also electroporation of T cells. So that was our story, how we became the clinical T-cell engineering experts. Now I would like to show you what our customers and collaboration partners did with our systems. So this here is an established track record for our CliniMax Prodigy T-cell transduction system and also the T-cell engineering system. So what you can see is that the first publications actually reach back to 2016, which was the year when we actually released the CliniMax Prodigy T-cell transduction process. And of course, the first publications were focusing on generation of preclinical data and also on manufacturing optimization. Now, from 2020 on, we can see already first clinical trial results. And the products used in that clinical trials were actually manufactured with our systems. And last but not least, the last two publications are actually showing really impressive results with our CliniMax Electroporator and also our T-cell engineering. And on my last slides, I will actually dive into that and show you some of those results. So how can we, with our experience, help you on your way to your engineered T-cell drug product? So first of all, we are always trying to offer research workflows that can be easily translated then into clinical. So in the example of the CliniMax Electroporator, you can start with the test cuvette adapter and small-scale optimization experiments, and then translate that right into the clinical setting and scale up with the same device. Once you're ready and you really want to move into clinical, we can support you with regulatory documents. We offer a device qualification package. We also offer several trainings, not only for manufacturing, but also for IPC and QC. Of course, for cell manufacturing, we're offering our instruments, but also we have all the consumables available that you need for manufacturing. And we also are already offering a wide portfolio of IPC and QC products. Today, due to time limitations, I would like to focus on the cell manufacturing part. So we have talked about the Clinimax Prodigy platform now for quite a while. For the ones that don't know it yet, I would like to introduce you to the whole platform. So the Clinimax Prodigy, as mentioned before, was launched in 2012, so maybe some of you are aware. This device can be used for automated and closed cell processing, cell separation, activation, transduction, expansion, and harvest. Now, this year, we launched 2 more modules that can be combined with the Clinimax Prodigy. One is the Clinimax Formulation Unit, which now also offers cell formulation and final fill. And, of course, we launched our Clinimax Electroporator, which allows cell electroporation, also in an automated and closed fashion. And both modules can be combined with the Prodigy to serve your exact needs. Let's have a look at some technical details and the key benefits of the platform next. So the Clinimax Prodigy contains a peristaltic pump and valves, and they control the liquid flow within the tubing set. Then the user can control the process via the touchscreen and the GUI. Additionally, the Prodigy contains a magnet unit. That's where the separation of cells takes place. There is a barcode reader to scan all the different consumables that are being used during the manufacturing, and also a sealer to remove bags and also sample pouches. The real heart of the Clinimax Prodigy is the centrical unit, which contains the chamber of the tubing set, and within that chamber the cells are being processed, washed, centrifuged, labeled, also cultured. So this is really the heart of the Clinimax Prodigy. The Clinimax Formulation Unit can be placed at the lower part of the Clinimax Prodigy and then enables the final fill of the cell product. The Clinimax Electroporator is placed on the left side of the Clinimax Prodigy and contains different bags to separate the cells and the nucleic acid until the pulse is performed in the electroporation cuvette. So what are the key benefits of the platform? First of all, you can benefit from several already clinical proven applications. And within that applications, there are definitely a lot of configurable parameters to keep flexibility so that you can transfer your protocol on the device. Then, since it's an automated and closed system, you can reduce cleanroom size and also staff number, which will reduce your costs. And also the device is able to support you from the discovery phase to the commercial scale, which is again decreasing your costs. Then, since it's a clinical proven system, you can minimize your time to market and just benefit from the system. And last but not least, you will really benefit from the consistent product quality. So since it is an automated system, you will always get the same quality, and all the different information gathered during the manufacturing run are tracked in a log file. Since the Clinimax Electroporator is quite freshly released, I would like to show you some quick facts. So first of all, it allows automated and closed electroporation, which is integrated into a regular Clinimax Prodigy workflow. The tubing of the Clinimax Electroporator can be sterilely connected to the tubing of the Clinimax Prodigy and therefore forms one big closed system. Electroporation of large cell volumes is enabled, and the Clinimax Electroporator is controlled by the Clinimax Prodigy. It can be placed right next to the Clinimax Prodigy due to its small footprint. And as mentioned before, upscaling and translation is easily enabled with the Clinimax Electroporator. The Clinimax Prodigy Tcell engineering is the perfect choice for your clinical scale Tcell electroporation. Within the process, the T cells are separated from the starting product and then activated. Then you can decide to either go for viral transduction or for electroporation. Or you could also combine both, so viral transduction and electroporation. After that, the genetically modified T cells are maintained in a culture and start to expand and can then be harvested once ready. So all the consumables for this workflow are available from Miltenyi Biotec. So we are providing the tubing sets and accessories, the separation reagents, the medium and buffers, as well as the cytokines and activation reagents. So what are the key benefits of the T-cell engineering application? First of all, it's really flexible to allow your process optimization. It allows different T-cell separation strategies, offers a customizable culture protocol, and gives you full control over the electroporation parameters. Then it's translational, so you can easily start with a small-scale test cuvette adapter and then, once ready, scale up with the T-cell engineering. Then it's gentle to the cells. The electroporation itself goes quickly, and then the cells are directly transferred back into cultivation. And last but not least, it's smart. So the cells and the nucleic acid are mixed directly before the electroporation pulse to avoid nucleic acid degradation. So having touched the subject of flexibility, I would love to introduce you to our activity matrix, which makes customization of your cultivation protocol really easy. So all these puzzle pieces that you can see here are actually activities, and they can be scheduled within the activity matrix to your liking. So you can plan exactly which activity you want to take place when. And you can choose from transduction, feeding your cells, medium exchange, stimulation, volume reduction, and so on, and really customize your protocol. Also, what you can see is that the electroporation is actually an activity in the culture, and you can really schedule it whenever you want it. So how exactly does it work? Your cells during culture are in the chamber of the tubing of the Clinimax prodigy, and once the electroporation is supposed to take place, the cells are transferred to the tubing of the Clinimax electroporator into the bag, which is currently marked in orange. Then you add your nucleic acid to the bag, which is now marked in turquoise. The portion of the cells and the portion of the nucleic acid are then mixed right before the electroporation pulse. And once that is done, the cells are transferred back into the culture where medium is already waiting for them. When thinking about optimization of the electroporation itself, there are several factors to consider. First of all, the question of which cells do I want to electroporate? And of course, which genetic information do I want to use? Then, what is my main goal or aim? So do I want to have a high number of transfected cells? Or is my main aim to get a high concentration of the transferred nucleic acid into one single cell? Or do I want to maintain a high viability after electroporation? And based on that main aim, you can optimize your parameters. And the parameters would be, for example, cell concentration, nucleic acid concentration, and then last but definitely not least, electroporation parameters. For example, electroporation parameters include voltage, and I think we all know how electroporation works. So you're applying a certain voltage to your cells, which then drive pores into the cells. If you're applying a very low voltage, you don't get any effect. If you apply a very high voltage, your cells die. So you need to find really the right balance to create pores but not kill your cells, and that is definitely not easy to do. And this is exactly why the Clinimax Electroporator is the perfect choice, because it really gives you full control over the electroporation parameters so that you can really optimize your protocol. So you can either work with one pulse or two subsequential pulses. And for each of these pulses, you can choose the voltage, the length, the mode, the polarity, and the burst length. And at the bottom of this slide, you can just see some pulses as an example that can be achieved with the Clinimax Electroporator. It can really go from a very simple single square wave pulse to a really complex double square wave pulse, which includes bursts and a bipolar pulse. So with the Clinimax Electroporator, you really have full control. So if we are thinking about what factors influence the electroporation efficiency, we talked about cell concentration, nucleic acid concentration, electroporation parameters. Also, the electroporation buffer plays a major role, of course. We are trying to avoid cell death, and we are trying to avoid nucleic acid degradation. And the Clinimax Electroporator offers all that. You can easily choose your cell concentration. You can also adapt your nucleic acid concentration. As you just saw, the electroporation parameters are really in your control. You can choose them and optimize them. Then when it comes to the buffer, we have worked a long time to optimize our CliniMax electroporation buffer. The whole procedure is really gentle. The cells are quickly electroporated and then guided back into culture. And last but not least, we're really trying to avoid nucleic acid degradation by mixing cells and nucleic acid right before the pulse. So all of these really important factors are granted with our system. So in the last part of today's webinar, I would like to focus on some really impressive publications from two of our collaboration partners. One was from the group of Boris Fese from Hamburg, and the other one from Tonika Thom from Freiburg. And both of them successfully established a protocol for our CliniMax electroporator and successfully manufactured gene-engineered T cells. The first approach was about knocking out the T-cell alpha beta receptor and transducing the T cells with a CAR to make them appropriate for an allogeneic approach. The second approach was knocking out the CCR5 in CD4 positive T cells to make them resistant against HIV. And now in the following slides, I'm going to guide you step by step through the data. So first of all, I would like to show you some data from the separation. So both groups separated T cells. And what you can see is that they had a really high T cell purity after the separation. Then both groups activated their T cells with our MaxGMP T cell transact. And in the one group, they used the microscope from the CliniMax Prodigy. And what you can see is that after activation, the T cells nicely start to cluster, which is a sign for activation. The genetic engineering is, of course, the most interesting aspect here in these publications. So let's have a look at the TCL alpha beta knockout CAR T cells first. They were using viral transduction to introduce the CAR, and you can see nicely in the upper graph how well that worked. And then they knocked out the T-cell alpha beta receptor with using TALENs, and you can also see that the indel rate was quite high and also the knockout efficiency was quite good. In the second publications, where they knocked out the CCR5, they also used TALENs to knock out that receptor, and you can also see how nicely that worked. They additionally also looked at the monoallelic and biallelic knockout. And what I found really interesting is that actually most of the cells have a biallelic knockout, which is of course exactly what they wanted to achieve. After the genetic engineering, the cells are expected to expand, and that is exactly what you can see here. So the cell counts after the electroporation drop for a very short period, and then the cells really start to expand. Same trend is visible for the viability. So shortly after the electroporation, the cell viability drops a little bit, but then afterwards the cells really recover quickly and then maintain a really high viability. In both publications, the groups also looked at the preferred T-cell phenotype, which is usually a memory T-cell phenotype. And in both publications, they could see that those phenotypes actually dominate in the final product. And last but not least, it's always very exciting to check whether the cells are really functioning as expected. So that's exactly what the two groups did. They did some functional assays as a last part in their publications. So the group with the TCL alpha beta negative and CAR positive T cells checked for cytokine secretion. So they mixed their CAR cells with the corresponding target cells and then analyzed whether the CAR cells still secreted cytokines even without the T cell alpha beta receptor. And as you can see nicely, that's exactly what they do. So when confronted with a JAK1 cell line, they nicely secrete cytokines. Then the same group also checked for killing capacity. So again, they mixed the T cells are beta negative, CAR positive T cells with the corresponding target cell lines, which would be the NALM6 and the JAK1. And what you can see is a really nice rate of cytotoxicity, and not for the negative control. Then, in the case for the CCR5 CD4 positive T cells, which should be resistant against HIV, that group also checked for cytokine secretion first, so that checked whether the knockout cells can still produce cytokines, and apparently they can do that. And last but not least, the most exciting part actually is the question, of course, can the HIV still bind to the CCR5 negative T cells? And they really nicely showed that the HIV cannot bind anymore, and that the cells are really functioning as expected and are resistant against the HIV. So really nice and impressive results. And hereby I would really like to thank our collaboration partners for these really two impressive publications. So we would be very happy if you would choose us as your trusted partner in your cell and gene therapy journey, and if you would benefit from our experience. Thank you a lot for your attention, and I'm now really looking forward to the panel discussion.
Thank you. Great presentation there from Melanie. So now let's move on to the panel discussion. So we'll welcome Kunal and Stefan. I hope you're both ready to answer some questions.
Yes.
Brilliant. So the first question I've got here, I think this one's probably one for Stefan. What are the challenges of development of clinical application for T-cell electroporation?
So I think the main challenge is that in the end you need a really robust process, and this should also include that you consider the donor variants. So this means that you really have to test many different samples to see what results you can achieve reproducible. And of course, this can be done in small scale and in part, but some of these experiments also have to be done, of course, at scale. So therefore, you have to also consider quite some time to set up these processes before you then can start really with starting with the clinical trial.
Absolutely. And another one for you here, Stefan. Electroporation, what are the advantages of electroporation in comparison to viral strategies?
So, okay, of course, the viral transduction, as Melanie mentioned in her talk, is well established and is used in the clinic routinely. And one advantage of the viral transduction is that you don't need any additional instruments. But on the other hand, the viral vector is quite costly. And for some applications, it's also difficult because it takes long development times if you need a new viral vector, or if you would think, if you're thinking about, for example, having patient-specific viral vectors. So this is, of course, one advantage of the electroporation, that it's more flexible, so it's easier to exchange the nucleic acids as compared to exchanging a viral vector. Another advantage of electroporation is that you can do additional engineerings, for example, doing knockouts of several genes, or combining knockouts with knock-in, which has also the advantage that you would get site-specific integration. And typically, it's easier to establish the electroporation as setting up the workflow for the transduction, including the viral vector production.
Absolutely. And this one, perhaps more one for you, Kunal. What approaches would you, or what areas would you say electroporation would be most valuable for within the cell therapy field? Kunal, you're having a bit of technical difficulty? I think you might be on mute, Kunal. Oh, well, never mind. Well, we'll go on to another question, and hopefully Kunal will be able to join us. So perhaps one here. What are some of the challenges when working with an electroporator?
So I think the first challenge, of course, you will have is to find the optimal pulse conditions. So as Melanie pointed out, you have to balance the cell survival and the transaction efficiency in the end. And of course, besides the pulse itself, you can also influence the outcome, for example, by the nucleic acid or ribonucleotide protein complex concentration, which also plays a role. For example, the amount of foreign DNA, if you are talking about plasmid delivered to the cells, can induce innate immune reactions in the cells and killing the cells just by the delivery of the foreign DNA, independently of the stress you induce by the electroporation itself. And even if you do this in small scale, then if you're thinking about clinical applications, you have another challenge in scaling the system up. So if you remember this typical electroporation processes, you're using 20 microliters or 100 microliters for the electroporation, so manually filled cuvettes. So if you need billions of cells, this would mean a lot of manual work and take a long time. So therefore, it's really crucial to have an automated system that can do this job and support you in setting this up, and to allow you to do this also in a GMP compliant manner.
Absolutely. And what would you say are what are the most commonly used gene editing approaches, and what are some of the advantages and disadvantages?
So of course there are different goals you can tackle with gene engineering processes. So like Melanie mentioned, you can do random integration by the so-called transposases. So they will introduce gene of interest, having the transposons flanked to the gene of interest randomly into the genome. So like the viral vector would also do. If you want to modify the cells, for example, by knocking out, then you would use site-specific nucleases. So classically, this would be done by zinc fingers or talents. So these are, again, well-established technologies which have also been used in the clinic already. However, the design of these nucleases is more complex, so it takes a longer time. And it's not easy to be performed. Nowadays, most of the people are using the CRISPR-Cas9 system because here it's much easier to design the target of interest because you simply have to adapt the guide RNA used. And you can also test many different guide RNAs and optimize it for optimized specificity. And if you do the knockout, of course, you can use the same approach to do knock-ins. So then really to integrate the gene of interest site-specifically, for example, exchanging the endogenous T-cell receptor by a new cancer-specific T-cell receptor, and then keeping the control of the cells on the gene, so in this case, the T-cell receptor active, so that the cells can adapt to the amount of antigens they see.
Great answer. Thank you, Stefan. We're working on getting Canal back. We think we may have resolved that problem soon, but I've got another question for you here, Stefan, actually. What would you say are challenges of development or translation of protocols into the clinic?
I think the biggest challenge for most of the customers is to get the process GMP compliant. So typically the processes are well established on a research scale, and the scientific work invested at this point is usually huge. So there's a lot of knowledge at this development stage. They have well established small scale processes with a lot of manual handlings. But if you have to do this routinely every day and also robustly so that you really can treat patients, the manual processes are most of the time not sufficient, or it would be too time consuming and too expensive. And the automation which is then needed to really translate the process is often a big challenge. And of course, you can start automating the single process steps and then combine the different process steps. So that's one approach many people are taking. And our approach had been that we would like to integrate all the different process steps in one system, that you really have one system which can cover the whole process. And now with the CleanMax Prodigy also including the Electroporation to ease this transfer from the small scale to the large scale process, and then also to be GMP compliant in the end.
Brilliant. I've got a bit more, some more specific technical questions here. Is your Electroporator integrated with the Prodigy, or do you do a manual transfer of bags between the instruments?
No. So as mentioned, it's also integrated. So basically, you can connect the Electroporation tubing set to the tubing set on the CleanMax Prodigy, and thereby you have then one integrated tubing set, and the cells are automatically transferred from the Prodigy to Electroporation. And then after the Electroporation, are also automatically transferred back to the culture chamber. So you don't need to transfer the cells from one system to the other, for example, using bags. So this is all automatically performed. And also the software is run on the Prodigy. So you have one software, one process controlling the complete production process, including the Electroporation.
Excellent. And I think we've got Kunal back on now, so that's great. So Kunal, I did have a question for you, which was what areas do you think Electroporation is most valuable for within the cell and gene therapy field?
Yeah, hi. Sorry, apologies for the technical difficulties earlier. But yeah, so I would say Electroporation, and Stefan kind of touched on a couple of these, but it's definitely a gene editing strategy that's really opening up the possibilities of what we can do now with our various gene editing strategies. The ability to add knock-in and knockout capabilities in conjunction, I think, greatly opens up the different types of therapies that we can produce, whether it's enabling the allogeneic space or even other complex modifications such as doing a knockout followed by even like an AAV transduction for targeted gene insertion. I think there's a lot of opportunities here that is really going to open up the field. And it's specifically when it comes to what type of cell types that it's enabling us to work on. I know traditionally viral vectors are used pretty extensively in the field right now. However, viral methods don't always work efficiently with all cell types. I know there's now a growing field right now in NK cells, where viral methods haven't worked in the same fashion as they do in the T-cell space. And here, I think Electroporation is really opening the doors for CAR NKs and other gene-modified NK cell therapies, as well as the hematopoietic stem cell space. I think there's a lot of interest now in the HSC space to allow for modification of these cells for various regenerative medicine indications. So I think with those combinations, the Electroporation really is opening up the doors for a lot of opportunity that wasn't there previously.
Absolutely. And I don't know, Kunal, if you want to field this one as well, what is the current knock-in performance?
Yeah, so with knock-ins, specifically with, the Electroporator, it really depends on what you're trying to knock in. Are you looking for a stable or a transient expression? If you want to integrate, large plasmid DNA, generally, the construct size and the number of base pairs is larger. And this inherently would require higher voltage differentials to generate a sufficient pore size. To be able to get this much genetic material into the cell, and then that is associated with higher cell death. So some of that it really depends on what your specific construct is, or if you're looking at mRNA for a transient transfection, Generally those are a little bit smaller and won't require as harsh of a pulse to get a high transfection efficiency. But in the end, especially with the way that the Miltenyi platform is, it's very customizable, where you can really fine-tune pulse parameters for your specific, gene editing strategy, and really play with maximizing transfection efficiency versus, cell death, and you really got to find that balance with your expansion potential of your cells as well. But in general, with knock-ins, I mean, you can get a pretty decent efficiency comparable. Maybe not quite as high as viral methods currently, but still somewhat comparable, I would say, and it's only improving right now as we get more and more experience.
Absolutely. And I don't know if you want to take this one, Stefan. How exactly does the Prodigy mix the cell suspension and nucleic acid prior to electroporation?
Yeah, that's a very good question, because of course this also has to be done automatically. And for me, mixing, there are two aspects. So the first is the mixing ratio, which needs to be accurate so that you really deliver the same amount of nucleic acid to the cells. And of course, the mixing has to be also done homogeneously. And we have included a microfluidic device in our electroporation cuvette, which basically cover both questions. So we have two microfluidic channels which define the mixing ratio of the nucleic acid to the cells. So this is accurately done because it's basically fixed by these microfluidic channels. And then we have a wiggling structure which mixes the cells and the nucleic acid just before they enter the electroporation cuvette, so that we really have a homogeneous mixture which is then electroporated. And it's also important that you do not have to pre-mix the cells with nucleic acids, especially if we are talking about RNA electroporation. If you mix the cells with RNA, the RNA gets immediately degraded, so this would not work in large scale. There's another difference between the manual handling and an automatic handling. So therefore, you really have to do this mixing also in an automated fashion just before the electroporation, and this is done by these microfluidic channels which are just in front of the electroporation cuvette, and then the cells immediately get electroporated so that this RNA has no time to get degraded until it's electroporated.
Excellent. And I've got another technical question here. Not sure who wants to answer this. For the electroporation unit, what capabilities are possible regarding the pulse applications? So, for example, what pulse magnitudes, durations, waveform shape, and pulse numbers are possible to program into it, or is it pre-programmed into the protocols? So, sort of Tcells, for example.
Yes. So in general, the protocols are not pre-programmed, so the customer is free to program the pulse scheme on his own. So he can flexibly adjust the pulsing parameters. The system offers two square wave pulses, one high voltage pulse for opening the pores, and then typically a second low voltage pulse is applied to ease the transfer of the nucleic acid or the RNPs into the cells. And you can choose the voltage as well as the time for both pulses. And of course, you can also do just one pulse if this is needed. But especially for the T cells, we have seen that it's much more efficient to have this two pulsing scheme. And in addition, we have the possibility to apply so-called interrupted or burst pulses, so very short pulsing schemes, which we have seen that in some instances this really improves the survival, and the effect have been even stronger if we do a bipolar pulse. So having this short burst pulses, but then change the polarity after each small burst pulse applied to the cell. This has also been advantages for the cell survival. So with these additional parameters, you have more possibilities to tailor the process, as also Kunal mentioned, whether you are going for higher cell survival or higher transfection efficiencies. So here you can also do some more fine tuning.
Yeah, sounds like there's a lot of flexibility there. I've got another question here. What is the largest volume of cells that can be effectively electroporated? If you want to jump in here, Kunal, or?
Yeah, I mean, Stefan could probably know this one a little bit more than me. To be honest, I always work on the lower end of the volume space, mainly because as long as you can get a good transfection efficiency, and we've done studies where we've worked with a range of cell concentrations in which we've seen very similar, if not equivalent, efficiency. And even cell concentrations that are a little bit higher, even, like 50 million cells per milliliter cells. And what's great about that is if you can minimize the volume space that you're electroporating, you minimize the amount of electroporation pulses and the time that the cells are, in the electroporation solution, and you can get them, back into media in a shorter duration. But saying so, I think the Miltenyi electroporator is a pretty flexible in that you can electroporate a fairly large amount of volume. I don't know, Stefan, you would know the spec, I think, exactly a little bit more than I do on the upper end.
Yes, so I can add this. So basically, you can electroporate up to 200 milliliters of cells mixed with nucleic acid. So according to the mixing ratio, this would be 157 milliliters of cell, and then you add nucleic acid, and then you come up to 200 milliliters of cell suspension, which can be electroporated. And typically, we go up to 5 times 10 to the 7 cells per milliliter. So as Kunal pointed out, typically we try to electroporate high cell concentrations. So thereby you can electroporate quite a decent number of cells, even if you are talking about really high cell numbers.
Great. I've got another size-related question here. The size of the bioreactor for the Tcell is quite small, says this listener. Are you planning to develop some larger-sized vessels?
Maybe I can take this. Yes. So we are working on a bigger chamber, which would cover then nearly 3 times the volume as compared to the current chamber. And this is planned to come next year, basically. And we will also have electroporation processes, which then would use the large chambers, of course.
Great. And if you do process large volumes with the electroporator, like the over 100 mils that was mentioned in the presentation, how do you ensure that the performance doesn't decline over time, for instance, by cellular debris accumulating on the electrodes? Kunal, do you want to jump in? Oh no, Stefan, sorry. Go ahead.
So the electrodes are gold coated. So thereby we ensure that, because that's the main problem if you are thinking about the classical electrodes, that they are typically aluminum based, and of course then you would destroy the electrodes after a few cycles of electroporation. So therefore we have chosen titan based material, and this is then gold coated to really have an inert surface of the electrodes. And this also helps, of course, that only a few cells or cell debris can attach to the surface material. And if you stay in the cell concentration range as we recommend, we have not seen that this impairs the cell debris on the electrode surface would impair the electroporation performance. So we see that electroporation performance is similar for the first electroporation as well as on the 20s or 100s electroporation pulse.
Brilliant. And if processing more than one donor at the same time, can the EP unit be moved across Clinimax Prodigy to reduce the processing time? If you'd like to have a stab at that one.
So typically, these systems are installed really as a system, and of course, as the software runs both instruments, it's not possible to exchange or remove the electroporator during one process. So you have to keep the system as a system together. And typically, if you're talking about clinical production, you would qualify the system also as a system, and then keep the system together because they have been qualified and tested. So therefore, you would not exchange the instruments during the process runs.
Right. I was just thinking out loud while you were mentioning that. I can just hear my quality team and validation team screaming at the thought of that, just because of the qualifications of the instrument for clinical use.
Brilliant. Well, I've got another question here. Is it possible to work in classroom B with our closed Prodigy bioreactor according to GMP, or is it necessary to work in classroom B?
So I think for that, some of it will depend on your local regulations and what the treatment specifications are, right? If it's at least in our clean room spaces at the San Jose site, we're maintaining our clean rooms as I believe a Class B background. Whether or not that's necessary for your specific program, I think is going to depend on the classification or category of how your treatment is seen by the regulatory agency. There are cases where you could do processing, minimally manipulated cells out in a Class C, but it really just depends, I would say, on your specific therapy.
Brilliant, thank you. And I think we've just got time for, we could squeeze one more question in. It's a quick text-based one, so perhaps one for Stefan. What is the size of the cuvette, or the electrode gap, that is compatible with the system?
Yes, so the electroporation distance is 0.2 millimeters, at least for a Tcell, so this has been optimized. And the electrode, the cuvette covers about 650 microliters, which then can be electroporated per cycle. And after the electroporation, the cells are pushed back, and then the next cycle can continue. So thereby we are cycling all the cells for the electroporation until the whole cell suspension has been processed.
Brilliant. Thank you, Stefan. Thank you very much, both of you, for answering those questions. So all that's left is to thanks once more to our presenters and panelists, and thank you to the audience for listening. We hope you'll join us again soon. Goodbye.
So we would be very happy if you would choose us as your trusted partner in your cell and gene therapy journey, and if you would benefit from our experience.